Joining us now from IDBS is Nick Fuller, Head of Product Enablement. Analytical method development is often slowed by fragmented systems and manual handoffs, increasing compliance risks. A unified digital approach streamlines method design to execution with structured data capture, protocol-driven assay setup, and traceable workflows under GXP. This presentation shows how integrated platforms enable visibility, version control, and performance insights to accelerate development and ensure data integrity. Welcome, Nick.
Thank you very much, Perry. So as Perry mentioned, we're here to talk about bridging the gap between your method design and the execution in analytical development. Now, IDBS is a software vendor that's been working in the life sciences industry for 35 years, mainly within GXP environments, and all of our software is designed to work in both GXP and non-GXP. And we are a Danaher company, so we're part of Danaher alongside other operating companies that we'll see in just a moment. Danaher, of course, has a number of companies across instrument vendors and other life science software companies, such as Gene Data that we've heard from today. This gives us a unique access and partnership with these other operating companies, which makes us a trusted partner in these areas. Some of IDBS's credentials are shown on this slide here. So we've been around, as I said, for 35 years. 75% of the top 20 biopharmas are using our software. We've had top industry awards, highest rated customer satisfaction across scientific lab data platforms. GXP excellence, as I mentioned, it's sort of part of our DNA, part of our culture around compliance, and of course, quality standards. So we hold our own standards in a lot of the areas around SaaS software deployment and compliance. Moving into the agenda for our talk today, we're going to look at the lab loop. So from planning, executing, and gaining insight on your data within the IDBS Polar software platform, optimizing a platform method to quantify a new protein, executing the method and obtaining your results, and then completing that lab loop through data-driven insights. So all within one platform to increase data integrity. It's all fair data ready for your AI ML use cases in the future as well. So I'll dive into the lab loop. This is how we describe the operations going on within our customers' environments, So we have the execution of the work, where the IDBS platform contains tools for requesting, scheduling, and sharing work for collaboration. We have configuration of workflows, a library of examples or accelerator workflows, instruments for integration, our chemical design and reaction planning, through to the data capture and storage. So we have experimental write-up, version control, and sign-off, our raw materials and recipe management, our sample management and genealogy, through to gaining those insights and getting that data out of the platform. So as I mentioned, our AI-ready fair data store with contextualized data across experiment, product, and process, and then our advanced analytics and insights, which really do allow for those more AI ML use cases going forward. Now that lab loop, we know, could operate faster and more efficiently. So that's IDBS's mission, to get those therapeutics to market faster by empowering and accelerating that lab loop. So why does Polar from IDBS as a single software platform matter to our customers? Well, we've heard it in a number of these presentations so far today, that often our customers are living in a world of siloed data. Lots of systems with disparate data structured differently with very little standardization or interoperability. What IDBS Polar aims to do is address this disjointed, siloed system and bring that data together, because at the moment with these disjointed systems, it causes delay, inefficiency, and it risks your scientific decision making. One mistake, one issue with any data integrity, you have to start again. It wastes time. You have to go back, root cause analyses, figure out where you went wrong. Through the IDBS Polar platform, we bring all of that information into one system. So you get your experiment, your product, and your process understanding all in one place. So we remove that risk, we remove those silos, and we allow you to gain your operational insights where you can make decisions quickly off of the data that's been captured. Looking at the data journey around the lab loop and applying that within IDBS Polar to our method development, optimization, and execution. Here you can see as that lab loop moves around from developing our method, so designing our method, choosing certain critical process parameters that we'd like to monitor and control, optimizing that method to ensure we're getting the best out of it for reproducibility, to ensure that we are able to validate that method also, executing on that method with some known samples, and then with some unknown samples, of course, using our calibration curve to perform a linearity assay. We then get our suitability insights for that method, so we can draw from the system insight into how suitable that method is for that particular protein that we are looking to quantify. This aligns with accelerating the lab loop through IDBS Polar to give greater value to our customers and get their drugs to market faster. So I'm going to show you now a demonstration of the platform and showing you how we manage methods and how we bridge that gap between method design and execution. So here I have a method definition within the Polar platform. Now the method definition is, for all intents and purposes, a multi-dimensional spreadsheet where I can specify all of the key parameters for my method, give my method a unique identifier that is immutable, as well as a unique version. Now, the important thing about the version of the method and the fact that it is unique across the system means that it aligns with things like ICH Q14 and Q2 revision 2. Here I can specify the types of inventory I'll need, so my chromatography column, what I'm going to need with regards to my buffers for my HPLC method. I can set up my different critical process parameters here. I can define how that process is going to look. These methods then have business rules built in for exceptions and when things deviate. If I have a crude sample that requires centrifugation, or I want to incubate it in a water bath before I perform my method, I can. Here I can set up my execution. So what type of HPLC? In this case, we're going to look at a protein A with a protein A column. And then here I'm really defining some of those critical process parameters around the column, the flow rate, anything that I might be interested in as part of this method. And then what I will do is execute this method, and then I can come back and I can tweak it, I can update it, I can upversion it, and then everything that I've done against that method is traceable throughout the system. So if I take a platform method off the shelf, I've got a new protein, I can then tweak that platform method to the needs of this specific new protein that I'm quantifying. You can see here we've got 5 minutes on, 10 minutes off with regards to our binding and elution buffers. And then we've got our reviewer summary here to check all of our mandatory fields, no exceptions, everything is good to go with regards to our method. Moving forward, we will save this method to our central method library within IDBS Polar, where, as I said, it's saved and versioned for execution. And now I'm going to execute against that method an HPLC. So I'm going to run my protein A for the quantification of my IgG antibody. I'm going to create a new experiment record using a template. From that, I'm going to pull in the details of my method. So once again, being able to define this method allows me to have the intent, the parameters, the version control, everything required for ICH Q14 and Q2 revision 2. And similarly here with my setup of my HPLC, I am able to enforce complete metadata alongside the inventory that I'm using. So which equipment, which buffers, which materials am I using? And this is all traced back through a usage log. So if I have any deviations, any issues, I can trace those issues back to specific equipment, specific materials that I've used. This is aligned with Alcoa principles around metadata and being able to reduce manual transcription. So here I'm pulling things in based on my method. So by querying the method from our centralized method store, I've actually pre-populated a huge amount of my execution workflow. So my workflow is populated based on what my method said that I should be doing. So our 5 minutes on, 10 minutes off with our binding and elution buffers has come from the method, for example. And then I can really execute against those parameters that I've set within my method. Here, I've got my sample set up. So taking, once again, the information from my method, I'm now setting up the specific samples I'm going to use. So I've got my unknown samples here, and I can choose which of those samples I'm pulling in from our request module, which is used for test requesting and analytical requesting from other teams. Here I've got any additional inventory I want to bring in. So if I'm going to do any preparations, it's any additional reagents that I need. Here I've got my setup of my standards. So I've got my standards of known concentration. All the information in blue you can see here is pulled from the method. So I haven't had to set any of this up manually. This is all based on the number of standards, the number of dilutions, the planned quantities that I would need to use, and then the actuals I've put in here for my execution. And once again, we can flag if we deviate or if anything is not as expected. These deviations get registered and are available in our Insight applications as well for review. Here I'm setting up my unknown samples. I've chosen in my method not to spike my samples, but if I wanted to spike my samples with something, I can choose to spike all or some of those samples. And once again, we're really just evaluating here, what did I plan as part of my method and what is my actual execution looking like? I'll skip the spiking here just because we haven't chosen to spike anything. Here we generate our run list for our samples. So when we run our HPLC, this is the run list that we're going to be using, and this is generated automatically by the system. Now I've set up my HPLC, now I've set up my samples, I'm actually going to execute the HPLC and get my results. So using those standards of known concentration and my samples of unknown concentration, I'll be able to execute my HPLC. And here you can see the same method details with the same method version. And then I have my mPower information. So IDBS Polar has a bidirectional integration with Water's mPower. So we can push information to mPower like a sample set, or we can pull back the result sets into our platform, as you can see here. From those mPower results, I can then draw up my standard curve from my standards, against which I'll be able to extrapolate the concentrations of unknown protein from my samples. So as I move through my peak results, obviously there's been some extrapolation from the peaks in order to help me do that. And then here we can see our standard curve. You can see the data related to our standards, and then our unknown samples below. And you can see curve fits generated within the system based on the results of each of those samples. Now, within each of these curve fits, I can open them up and I can knock out data points, which will modify the underlying calculation that's retrieving some of the parameters from those curves. Because, of course, from those curves, I'll want to be able to calculate things like R squared, perhaps. So here you can see I can knock out data points, knock them back in again. I can highlight parts of the curve if I need to. And then finally, performing a linearity assay, for example, I can see here the linearity across those samples from my HPLC execution. So we've developed the method, we've saved the method and versioned it, we've executed the method, and now we're going to learn how that method performed for my particular protein. So I'll use the insight capabilities within IDBS Polar. Because our data is structured, contextualized, it's fair, we're able to generate insight directly from that data. So I can see here across my runs, across bioprocessing, so it's not just specific to analytical, I can drill down into specific samples, I can access specific records directly from here to see what was done. Now, that brings me to the end of my presentation. I just want to summarize what we've seen. I know it was very quick, it was a lot of information. The key things are IDBS Polar brings together your experiment, product, and process data. This highly contextualized, precisely structured data is perfect for reuse. By being able to have a centrally managed method library that is versioned, we're also able to ensure reproducibility and consistency. We accelerate your high quality method optimization through this unified data platform. This drives quality and compliance against FAIR and Alcoa++ principles. And overall, we accelerate the lab loop from the design of your experiment all the way through to gaining insight and executing the next one. Thank you very much for your time today. And I look forward to hearing any questions that you've got.
Thank you, Nick. Great presentation. So I'm a CDMO. I have a number of clients. They all come to me with different method requirements and SOPs. So how can you help me with that with a single platform as yours?
Sure, excellent question, thank you. So it's a common problem. We work with a lot of the big CDMOs, so quite a few of the top 20 CDMOs work with us and their sponsors. So Polar Control is a part of the IDBS Polar platform that provides a centralized parameter and process repository. So this brings alongside the business rule management of our workflows within IDBS Polar and our focus on quality management, a much quicker and easier review process, the ability to monitor and track exceptions. So when you have different methods, different method versions, that centralized versioning and oversight makes it much, much easier. You can take your method, you can tweak it, you can up version it, and then you can use it for a different client's needs.
Okay, we have less than a minute, so briefly, the other big challenge that drug developers often face is I've got a variety of instruments, I've got systems, I've got document management repositories. How compatible are you with all of that?
Absolutely. And this is a problem that, once again, all of our customers bring to us. So we have a variety of integration approaches across our platform, from flat files, simple text file imports, data links where you can use SQL, JDBC connections, to our really feature-rich set of RESTful web services. So we have a huge number of well-documented public APIs for integration with our platform.
All right. Well, Nick, thank you for being with us today. Great solution. Nick Fuller from IDBS.